I have some 500 to 600 questions that are being repeated again and again in DNB, both DNB and AIIMS examination. First question is, what is a dietary fiber? Is it a monosaccharide, disaccharide, polysaccharide, or a non-starch polysaccharide? The answer is very simple. It's a non-starch polysaccharide. We have done this repeated times. But what is the difference between a polysaccharide and a non-starch polysaccharide? Now, in polysaccharide, we come across glycogen and starch, sometimes cellulose. Okay, glycogen and starch, these are completely hydrolyzed inside the body by amylase, and it breaks down to amylopectins and minor forms of carbohydrates. Now, if this polysaccharide was given as a dietary fiber, would it really help us? It wouldn't really help us, because all this is getting digested. So, we need to give something which is a non-starch or non-glycogen containing polysaccharides. Advantage of a non-starch polysaccharide as a dietary fiber is the glycemic index is really low. That means they are slowly absorbed over a period of time, very slowly, okay, so that your blood sugar levels won't shoot up. Now, giving dietary fiber with something will produce a glycemic index which is really low, and it will help blood glucose maintenance. In diabetics, someone is diabetic, will you give corn flour, corn soup? You will not give corn flour. Why? Because it is having a really high glycemic index. You give him cornflakes, immediately it gets absorbed. You give him white bread, immediately it gets absorbed. Same thing with sucrose. Now, sucrose and sucralose, what is the difference? This is fructose plus glucose, this is fructose plus galactose. What is happening here is sucrose will hydrolyze immediately to glucose and fructose, both will give a good amount of sugar, okay, glucose and fructose. Whereas sucralose, which is there in artificial sweeteners, what is happening here? It is not breaking down easily. That means its glycemic index is really low. So, you give that to a diabetic patient in his coffee, it will still taste sweeter, but it will not increase the blood glucose level as fast as sucrose. That is why sucralose is given. Okay. Now, this dietary fiber, this is like cleaning my car. Okay. Now, this dietary fiber, it can be either soluble or insoluble. Lignins are the ones which make it insoluble, okay, whereas pectins and other compounds will make it soluble. What is the difference? Insoluble will totally add to the stools. You want to produce bulk stools in a patient, you give him a dietary fiber which is rich in lignins. Okay. You don't want big stool quantity, but you still want the glycemic index to be low, you give something which is having a lot of cellulose and pectins. Okay. Now, dietary fiber is a non-starch polysaccharide. So, most of them it contains arabinoxylans, cellulose and many other components, dextrins, inulin, lignin, chitins and so on. Carbohydrates, how are they classified? They are classified into monosaccharides, disaccharides, oligosaccharides and polysaccharides. Now, monosaccharides can be either a aldose sugar or a ketose sugar. Aldose is a glucose, ketose is a fructose. What is the difference? It is having a CHO group, that is why we are calling it as an aldose sugar, and this is having a keto group, that is why it is called as a ketose sugar. Example is fructose. Now, disaccharides can be either reducing which is lactose and maltose, and non-reducing, which is sucrose. Now, the question they are asking on non-reducing sugars is, why are they non-reducing first? What is the reason? Why do they need to be non-reducing? They are non-reducing because their functional group is free. Their functional group is free. Now, the functional group in a sugar is either the aldehyde CHO or the C double bond O keto group. Aldehyde or a keto group. If this aldehyde or keto group you add alcohol to it, it becomes a carbohydrate. It becomes a carbohydrate. If you are adding an amino group and a carboxylic acid to this alcohols, amino group and carboxylic acid to this alcohols, it becomes proteins. And if you are just adding carboxylic acids to alcohols, it becomes lipids. Okay. Now, everything depends on these functional groups. The properties, all these properties are dependent on this functional groups. Okay. So, now okay, carbohydrate, what are the two functional groups? One is aldehyde, one is a keto group. Now, if we see structure of glucose, the aldehyde is present at the end, or carbon number 1, or carbon number 1, whereas if we see fructose, it is present on carbon number 2, it's present on carbon number 2. Now, there are so many disaccharides. Maltose is glucose plus glucose, is glucose plus glucose. Lactose is glucose plus galactose. Okay. Next, sucrose is glucose plus fructose. Next, trehalose, insect lymph, when you spat that insect, white thing comes out, insect lymph, it is having a sugar that is trehalose. This is also glucose plus glucose. Now, two of these are reducing, and two of these are non-reducing, and two of these are non-reducing. One is reducing and second one is non-reducing. Now, these two are reducing. Why? Because glucose in maltose, it is 1,4. This 1, this is 4 hydroxyl group. This bond here. Okay. Suppose take this is as glucose only. Alpha 1,4 bond. Okay. Now, this alpha 1,4 bond, this functional group, it is busy in this bonding, but this functional group is still free. That is why it is a reducing sugar. Same thing with the lactose. Lactose is 4 epimer. Is beta 1,4 bonding. Okay. Why? Because galactose is a beta isomer, okay, or an epimer of glucose on the 4th position. Mannose is on the 2nd position. Galactose is on the 4th position. Now, that is why it becomes a beta 1,4 bond. Here also a aldehyde group is busy, but the other aldehyde group is free. That is why these two sugars, maltose and lactose, both are reducing sugars, or they have reducing properties. Now, if you look at sucrose, sucrose is having glucose and fructose, and this fructose is having on the 2nd carbon, and the bond formed in sucrose is alpha 1,2. The functional group here and the functional group here, both are busy. They are not free to cause any form of reduction. They are not free to form any form of reduction. That is why it becomes a non-reducing sugar. Same thing in trehalose. Glucose and glucose have alpha 1,1 bonding. Alpha 1,1 bonding. Both the aldehyde groups here are busy in functional group. That is why they become non-reducing. Now, what is the Why are we calling it reducing? Okay. We are calling it reducing because all these sugars, glucose, fructose, galactose, all these sugars are reducing because they will convert cupric ions to cuprous ions. They will convert cupric, they will reduce cupric ions to cuprous ions. Okay. Now, because this conversion is possible by these, they are known as reducing sugars. Now, which is the test we do? Which is the test we do? It is Benedict's test. What is the principle behind Benedict's test? Is formation of inidiols in alkaline medium. Formation of inidiols in alkaline medium. They are going to test you only on two tests if in case they want to ask a question on test in carbohydrates. Okay. One is Benedict's test, the other is Mollis test. Mollis test is a general test for identification of carbohydrates. Okay. What happens here is they form furfurols in acidic medium. They form furfurols in acidic medium. Suppose you add acid, sulfuric acid or nitric acid or HCl, whatever you add, carbohydrates will become furfurols. If it is a hexose sugar, it will form hydroxymethyl furfurol, otherwise only plain furfurols. Okay. So, Mollis test, furfurols is the compound that is formed and it is in the acidic medium, whereas Benedict's test forms inidiols which is in alkaline medium. Now, this will reduce cupric ions to cuprous ions, that is why it will give different colors. What are the 4 different colors that is formed in Benedict's test? Once rarely it was asked somewhere in some other test. If you remember this, we have read it in our childhood, LKG, VGR, okay? Violet, indigo, blue, green. So, 380 nanometers to 680 nanometers VGR we are having. Now, Benedict's test, B for Benedict's, remember like this, B for Benedict's, B for blue color. Benedict's test is, Benedict's reagent is having a lot of copper inside. So, the solution is blue. So, first the solution is blue, that means the concentration of sugar in urine is 0. Okay. Next, when you heat it, it becomes green. Okay. Concentration becomes 0.5. When it is yellow, it becomes 1. When it is orange, it becomes 1.5, and red means 2.0 or greater. Okay, red means 2.0 or greater. So, based on the color, we can say how much amount of sugar is present in urine. That is why it is also called as a semi-quantitative test. So, Benedict's test checks the reducing properties of sugars. Reducing sugars, monosaccharides are glucose, fructose and galactose. Disaccharides are lactose and maltose. Okay. Next is maltotriose. Polysaccharides, coming to homopolysaccharides means same amount, same units are there, monosaccharides, that is glycogen as well as starch, cellulose, and heteropolysaccharides which are having an amino sugar and a uronic acid. They are called as heteropolysaccharides or mucopolysaccharides. Now, a non-reducing disaccharide is, it is both sucrose and trehalose. The question should be in humans. In humans it is sucrose, if they ask in animals it is trehalose, or insects it is trehalose. Okay, coming to disaccharides, where is maltose present? It is digestion of amylase. Lactose is present in milk. Sucrose is known as cane or beet sugar, and trehalose is present in fungi or yeast, and also sugar of insect hemolymph. Now, sucrose is also called as invert sugar. What is invert sugar? They are asking common names: table sugar, invert sugar. Sucrose, it is made up of glucose and fructose. Now what I have done, I have put a monochromatic light, and this light is passing through this solution. Okay. If it goes to the right, then it is dextrorotatory. If it goes to the left, then it is levorotatory. Okay. Now what is happening here is because of the bonding, alpha 1,2 bonding, it is mostly dextrorotatory. Okay. Sucrose solution is dextrorotatory. If I hydrolyze it, if I break it, what is happening is fructose is having a strong levorotation. So the entire solution will start changing towards the left or levorotatory. So on hydrolysis, a dextrorotatory solution becomes a levorotatory solution. That is why sucrose is known as invert sugar. Okay. So coming to heteropolysaccharides, what are heteropolysaccharides? They are a uronic acid plus a amino sugar. Okay. All glycosaminoglycans, they are having a uronic acid and a hexose or amino sugar, except for keratin sulfate, which is not having a uronic acid. That is also a most common question in APPG. Which glycosaminoglycan is not having a uronic acid? Which of the following glycosaminoglycan is not having a uronic acid? So it is keratin sulfate that is not having a uronic acid. Okay. Next, mucopolysaccharide hyaluronic acid, where is it present? It is present in the vitreous humor. Okay. So glycosaminoglycans are complex sugars which are having this amino sugar and a uronic acid. Examples are hyaluronic acid, chondroitin sulfate, and so on. If you have to remember this, remember it like this: galactose amine. Sugar is galactose amine. Amino sugar is galactose amine. There are only two compounds which are having a galactose amine as their amino sugar. One is chondroitin sulfate. The other is dermatan sulfate. Chondroitin sulfate and dermatan sulfate. Chondroitin sulfate is having a glucose amine. Chondroitin sulfate is having a glucose amine. Dermatan sulfate is having glucuronic acid. Whereas dermatan sulfate is having a iduronic acid. So sugars which are having galactose amine or heteropolysaccharides which are having galactose amine or chondroitin sulfate and dermatan sulfate. Chondroitin sulfate is having glucuronic acid plus galactose amine, and dermatan sulfate is having iduronic acid plus galactose amine. Now why is this important? Because if suppose a dermatan sulfate is obliterated, okay, or dermatan sulfate gets accumulated, what are the sugars that we are going to see in blood and urine? One is iduronic acid if it is dermatan sulfate, and glucuronic acid if it is chondroitin sulfate. Okay. Next, coming to hyaluronic acid. Hyaluronic acid is the only one which is not sulfated. Rest all galactose amine sugars are sulfated. Glucose amine, if the sugar is glucose amine and it is not sulfated with a glucuronic acid and a glucose amine which is not sulfated, it is hyaluronic acid. Synovial fluid, vitreous humor, both places hyaluronic acid is abundant. Glucose amine and a glucuronic acid. Okay. Next, if it is sulfated glucose amine, okay, and a glucuronic acid, then it becomes a heparin sulfate. Okay. And if it is iduronic acid, it is heparin. Iduronic acid plus a glucose amine which is sulfated, it is heparin. Glucosamine not sulfated with glucuronic acid, it is hyaluronic acid. Glucuronic acid with sulfated glucosamine, it is heparin sulfate. Iduronic acid with sulfated glucosamine, it is heparin. And when coming to keratin sulfate, there is no uronic acid, only a galactose amine is present, or sorry, a galactose is present along with a glucosamine, sulfated glucosamine, then it is keratin sulfate. So, point to remember, if there is galactose amine, then it can be either chondroitin sulfate or dermatan sulfate. If it is glucuronic acid, then it is chondroitin sulfate. If it is iduronic acid, then it is dermatan sulfate. Okay. No other option, you will have to remember it. So, where is this hyaluronic acid present? Umbilical cord, synovial fluid and vitreous humor, a lot of it. Chondroitin sulfate, where is it present? Cartilage and bone. Dermatan sulfate, where is it present? Skin, aorta or blood vessels. Heparin sulfate, again in blood vessels. Heparin is present in mast cells. Okay, heparin is present in mast cells. Keratin sulfate is present in cornea and sometimes in cartilage also.